morning to all our viewers. On behalf of VIT Bhopal, I, Sanya, welcome you all to the third lecture of the planned lecture series of the Extended Science Week being celebrated in VIT Bhopal. Catalyzed and supported by National Council of Science and Technology Communications, NCSTC Department of Science, Government of India, and Madhya Pradesh Council of Science and Technology. In India, we celebrate National Science Day on 28th of February to commemorate the discovery of Raman effect by Dr. C.V. Raman. And today, it's an honor for us to have Dr. Satya Surabhi, a staff scientist at Albert Einstein College of Medicine in Bronx, New York, and she will deliver us a lecture on understanding autophagy in health and diseases. About Dr. Satya Surabhi, she's a staff scientist at the Albert Einstein College of Medicine in Bronx, New York, working in Professor Andreas Jenny's lab with a focus on developmental and molecular biology. She completed her PhD in biotechnology at Banaras Hindu University, mentored by Dr. Mosmi Mutsadi, where she developed a keen interest in the genetic and molecular biology aspects on neurodegenerative diseases, particularly the autophagy pathway. Her academic journey also includes a MSc in biotechnology from Maharaja Sayajirao University of Baroda and a PhD in chemistry from Banaras Hindu University. During her postdoctoral tenure at Northwestern University in Professor Ravi Alada's lab, she investigated circadian rhythms, further enriching her research experience in genetics and molecular biology. Dr. Surabhi has been recipient of a prestigious research grant from the American Parkinson Disease Association to study the role of NAMP protein in age-related neurodegeneration. Her extensive publication record in peer-reviewed journals showcases her significant contributions to understanding autophagy in neurodegeneration and the regulation of Notch signal. We welcome you, ma'am, today, and over to you for your lecture.
Thank you so much. So can you see the slides?
Yes, ma'am.
Okay. So first of all, I'd like to thank you for giving me opportunity to present my work here. And I'll start with, so as you already told that I'm working on autophagy process. So here in New York at Albert Einstein, what I'm trying to study is how, what are the different kind of autophagy process and how dysfunctional autophagy can affect our health and is related to disease like neurodegenerative disease. So before delving into my own research, I'd like to acknowledge the work of Dr. Yoshinori Osumi, who received Nobel Prize in 2016 in Physiology and Medicine. And before him, the autophagy process was not well known. So his work discovered the mechanism of autophagy, the gene related to this process, and how autophagy process is crucial for cell survival, especially under stress condition like starvational stress and oxidative stress and different kind of stress condition. So to start with a brief outline of autophagy process. So autophagy process is lysosomal degradation of intracellular material. So this is the lysosome, the most important organelle for autophagy. It has many acid hydrolase enzyme which degrade the misfolded protein or aggregated protein. So by doing this, it maintains the quality control of cell. Because a protein, when it's not functional or misfolded, it needs to be degraded. So it also degrades damaged organelle like mitochondria, you can see here, and pathogens which invade our cells. So this process actually maintain the quality of cell, and it helps in maintaining homeostasis as well as it helps in cope up with the stress. So always there is a low level of autophagy occurring in basal condition, but it gets upregulated when there is stress like nutritional stress or other kind of stress. So, when the autophagy research has grown a lot, and you can see from this diagram, there is like when autophagy is dysfunctional, it can affect many things in a cell and can lead to many diseases like cancer, neurodegeneration. It can also affect immunity. It has been manifested in many lysosomal storage disorder and diabetes, and of course in aging. So we know that when we age, there is decrease in autophagy process, and the cell cannot perform the degradation of waste. So my focus is in my lab is to study neurodegenerative diseases because neurons are more vulnerable to decrease in autophagy. Neurons do not divide, so they cannot dilute the toxicity. Coming to the how the actual process occurs in a cell. So I have shown here the starvational stress, which is the most extensively studied stress to induce autophagy. So it starts with the formation of a double membrane structure called phagophore, and these phagophore try to engulf the damaged organelle. Here is mitochondria and other damaged protein, and it grows this membrane and it form a double membrane structure. You can see here autophagosome, which is now containing this material. And these autophagosome fuse with lysosome, and this organ vesicle is called as autolysosome. And then these damaged organelle or proteins are degraded in the lysosome with the hydrolysis of lysosome, and then the the material is recycled to provide energy or to cope up with stress. So when you see with the transmission electron microscopy, you can see here the autophagosome. You can see here clearly the double membrane structure, which has, this one is mitochondria and the other part of the cytosol. So this process is the canonical autophagy process, which is very simple, and it degrade in bulk the cytosolic material. So I'll also briefly explain the endolysosomal pathway, where the endocytosis occur and it crosstalk with the autophagy pathway. So what happened during the uh, cytosis, there is like extracellular signal which through the receptor and the cell membrane inward invaginate, and then it mature to many different kind of endosome. So early endosome, late endosome, and then this late endosome finally fuses with the lysosome to recycle the receptor or to degrade it. So what happened, this process, the endosome mature and with different pH condition. And this pH is very important because it when it traffics some cargo. So depending on the pH of the system, the cargo destiny is determined. So if this pH is messed up, there are different diseases, like especially neurodegenerative diseases, which are known to occur when the pH is disturbed in the endolysosome. So you can also see this is the late endosome. So late endosome is also known as MBBs, multivascular bodies. So they have intraluminal vesicle where the cargo is trapped. So these process also intersects with autophagy. This late endosome can fuse with lysosome to form endolysosome, and then this endolysosome can degrade the waste material or can recycle the product. So in my talk, I'll be uh, uh, talking about endolysosomal pathway throughout. So when we started talking about autophagy, we started with the macro autophagy, which is called as MA, and is the canonical form of autophagy. So it occurs in bulk. Here you can see it, the double membrane structure that I have mentioned engulfs the cytoplasmic material and fuses with the lysosome. But this is not that simple. There are other forms of autophagy also, which are called as chaperone-mediated autophagy and endosomal macroautophagy. So what happened in chaperone-mediated autophagy? It doesn't occur in bulk, and it doesn't require the formation of double membrane structure that is called as autophagosome. So it's very selective for some protein which have pentapeptide motifs in their structures. So these protein are very abundant soluble protein in the cytosol. When these protein are recognized by a chaperone called HSC70, and then these chaperone present this protein to the lysosomal membrane, where these protein, when unfolded, they translocate inside the lysosome with the help of a lysosomal membrane protein known as LAMP2A. So LAMP2A is a splice form of lysosomal associated membrane protein. In human, there are like LAMP1 and LAMP2 is the very abundant lysosomal protein. So there are 5 lysosomal associated membrane protein. There is another form of autophagy which is called endosomal macroautophagy, which is both bulk and selective for this KFRQ motif containing protein. So what happened, this system now is engulfed inside the late endosome. When the late endosome is forming a intraluminal vesicle, these soluble protein are engulfed in that, and then it fuses with lysosome, and then the material is degraded. So these 3 kind of autophagy coexist in mammalian cell, and it is very important to note that when one form of autophagy is dysregulated or is non-functional, other form of autophagy get upregulated to compensate the process. So it's important to study all 3 different type of autophagy. In my lab, we are working on Drosophila model, also called fruit flies. So in fruit flies, this LAMP2A protein is not present. That means this CMA-like process doesn't occur. So there are only 2 type of autophagy process, endosomal macroautophagy and macroautophagy. While in mammal, all the 3 coexist. So in what we other members in my lab found that these type of autophagy process depend on the formation of multivascular bodies, and it depend on escort machinery. So when we delete escort machinery, this process does not occur. But the role of LAMP protein was not studied in Drosophila. So my work was to study what LAMP protein is doing and how it is important in Drosophila, endosomal macroautophagy or macroautophagy. So you can see here, this is a complex picture, but it shows very nicely how 3 kind of autophagy process cooperate, and when there is a dysfunction in the gene coding for this process, it can lead to multiple neurodegenerative diseases, among which Parkinson disease and Alzheimer's disease are the most frequent disease, which occur in elderly people whose age is more than 65 years most frequently. And there are mutation in gene, although there are different mutation, but the best known mutation is GBA1, which is a risk factor for Parkinson patient. So when there is a mutation in a gene called glycocerebrosidase, it result in more in the people to have Parkinson disease more than the normal people. So coming to how we study autophagy. So there are reporters which are known to label autophagism. So when we know the gene responsible for autophagy process, we can use them to study, to label autophagosome. So one of the gene is LC32, which is called as ATG8 in flies. So what happened, this LC32 decorate the membrane of autophagosome. So you can see here, and then when it degrade to lysosome, it get degraded along with. So we have a reporter where we have a GFP fluorescent protein fused with LC32. So now we can study autophagy in vivo and in different settings, like in disease condition or different. So you can see here, when in basal condition, this marker is not diffuse, while in the starved condition, when autophagy is upregulate, you can nicely see here punctate structure, which are the marked autophagosome. So this is the autophagosome containing the cellular waste. So this way people study macroautophagy. So what is how we study other form of autophagy? So I already mentioned that CMA and EMI degrade protein which have KFRQ motifs. So the lab, our neighbor lab in Albert Einstein, they have made a KFRQ based sensor to monitor CMA in mammals. And what they did, they made a fusion protein with KFRQ peptide, which is mCherry, and this peptide is photoactivatable. So it only fluoresces when it is photoactivated at 405 nanometer wavelength. So it get fluorescent, and then they change this protein, this labeling to both in fed and starved condition. And you can see here in fed condition there is diffuse staining, while in starved condition you can see punctate structure, which is most likely autolysosome or lysosome. So the difference is this kind of autophagy uh, required prolonged starvation. Here you can see 16 hours. And I forgot to mention that the macroautophagy is mostly peaked at 4 hours. So this is the difference. So what we did in Drosophila. So first I will try to explain to you why Drosophila and fly we study and why it is important. So fly has a life cycle of only 10 days. So start with the egg laying, you call embryo, and then first instar larvae, second instar larvae, then it pupate. So this is the developmental stage, which is within 10 days. So it has very low generation time. And then the other thing is the fly has eyes and wings that have similar genetic signaling pathways as compared to like human. So we can these pathway are conserved, and these part like wings and flies eyes, they are not important for viability. We can easily manipulate them, they will not die. So it's easy to see in vivo genetic manipulation. And the thing is like 70% of the disease loci are conserved between human and flies. So that makes flies awesome model organism to study and to do genetic manipulation. So what we did is, we make a KFRQ sensor in flies. So it was, we did a similar strategy that I have shown for mammalian system. So we cloned the peptide, which is KFRQ peptide with the mCherry, and that is photoactivatable. So in flies, there are systems where you can control the expression of a gene in a tissue-specific manner. So we choose a fly fat body that we extract from larvae. So why you choose fat body? Because fat body is analogous to mammalian liver or adipose tissue. So you can see here we cross two flies, male and female, and the male is carrying a activated protein called GAL4, which will bind to the UAS system, and then only this peptide will express. So we can control the expression of the gene. And then we took progeny larvae where we tracked this reporter. So we use fat body specific Galfor line, and we treat the flies for starvation to only 20% sucrose, and for the basal fed condition, we supplement them with the yeast. So when I say starvation, it is like amino acid starvation. that we have done. And then we chased the reporter for 4 hours and 5-hour post-starvation. What we found here is like this is the fat body of Drosophila larvae. And you can see here in the fed condition, there's like no punctate structure and there is only diffuse sensor. While in starved condition, then there is like autophagy, EMI is upregulated with prolonged starvation here, 25 hours. You can see very nicely punctate structure, which are autolysosome or which are lysosome, endolysosome. So these are the reporter I have mentioned that were very important to study autophagy and was made in one of the reporter was made in our lab. And then I will jump to the LAMP protein, which I mostly studied during my postdoctoral tenure. And what are the LAMP proteins? So LAMP are the lysosomal associated membrane proteins that I've shown. And what we have studied is like we found that LAMP is important in lipid assimilation and the acidification of endolysosomal system. And we also studied the role of LAMP protein in Parkinson's disease. So coming to the Drosophila lamp, so we found a homology between LAMP1 in Drosophila and human LAMP1 and LAMP2A. So Drosophila lamp has similar LAMP domain as of human, and it is also glycosylated. You can see here the red thing that is glycosylated. So what happened to the LAMP1 and LAMP2 in mammal? They are very abundant protein. They are almost they comprise almost 50% of lysosomal membrane protein, and they are glycosylated. So why they are glycosylated? To protect the lysosomal membrane from its own hydrolysis, so that the acidic environment inside the lumen of lysosome do not degrade its membrane. So LAMP protein protect them. So in study, they found that the LAMP1 and LAMP2 mutant mice, where like LAMP1 was mostly normal, LAMP2 mutant has some of the macroautophagy defect, and they also lack CMA because LAMP2A is a rate limiting step in CMA. So when there is no LAMP2A, that means there is no CMA occurring. And they have been manifested in a disease called Danon disease. So what happened in Danon disease is there is like accumulation of autophagosome, and then there is cardiomyopathies. But the double mutant, when they made a double mutant of LAMP1 and LAMP2, it was totally lethal. So they cannot get progeny. And what happened in LAMP1 in flies, it was totally uncharacterized. So I started with saying to understand the identity of LAMP protein in Drosophila. So what I did a simple dextran uptake assay. So dextran are the polysaccharide molecule which are taken up inside the cell with the process of endocytosis. So here you can see. And after 90 minutes of dextran exposure, it will flourish in late endosome and lysosome. It's already known. So I choose this molecule, a fluorescent molecule, and I expose Drosophila larvae, fat body, with the dextran. And then I co-label with the LAMP antibody that we made in our lab. And what I found that here you can see that both LAMP and dextran co-localize nicely after 90 minutes of exposure. That shows us that the LAMP protein localized to endolysosome as in mammalian system. In mammalian LAMP, it is known that it is localized to endolysosome. So I studied in a different way also to confirm my results. So what happens, so LAMP protein is synthesized in ER, endoplasmic reticulum, and then it goes to Golgi complex where it is modified and glycosylated. And then with LAMP carrier, it moves to either to late endosome directly or to the early endosome and then to the late endosome. So we have different markers to label this organelle inside a cell, like CX99A for ER, GMAP Golgi for Golgi, and RAP-GTPA is known to label endocytic machinery, and RAP7 is specifically label late endosome. So I did all this co-immunostaining with LAMP protein, and what I found that it co-localizes mostly in both fade and starve condition with RAP7 and cathepsin L. So these RAP7 is a late endosomal marker, and cathepsin is an enzyme that is found in lysosome. So it marked lysosome, which shows that the LAMP protein in Drosophila is also similar to the mammalian LAMP, and it localized to endolysosome. So what next I did was to make a mutant protein of LAMP, and we generated an allele called LAMP1 6.1 with the CRISPR-Cas technology. And what we found that these, when we mutate LAMP in flies, they have no problem at all. So they are happily surviving. They have no phenotype in wing and eyes, and I don't see any developmental defect. So how we measure developmental defect? We uh, uh, we try to measure here you can see that to pupae, how many embryo are formed and how many first instar and second instar, and what are the time period to develop from embryo to pupae. In this way, I measured it and I found that there was no developmental defect. So it was very surprising to us because LAMP1 has a very important function in mammal, and LAMP1 and LAMP2 double mutant is lethal. So it was surprising because Drosophila has only one LAMP, and that when we delete has no effect. But I see a very interesting phenotype. So I wanted to see whether LAMP has a defect in lysosomal biogenesis or endolysosomal system. So what I did, I labeled Drosophila larvae fat bodies basically with lysotracker staining. So this lysotracker dye is like protonated in a acidic environment and it fluoresces. So it can only label endosome, which are acidic, and lysosome, late endosome and lysosome. So you can see here, when in the fed condition, it's not likely that lysotracker will signal. But in starved condition, when autophagy is regulated, there are so many acidic autophagosome and other acidic vesicle. So it is expected that we will see lysotracker staining. And what we found that uh, in lamb mutant, we used two different lamb mutant, and we found that in lamb mutant, not only in the starved condition, but also in the basal condition, like fed condition, we see lysotracker staining, a lot of lysotracker staining. So this upper panel is a control fly that we commonly use as a control because there is no mutation in those flies. Only the eye is mutated, eye is white colored. So when we have lamb mutated, you can see here, there's so many lysotracker puncta. So we wanted to check whether it is an artifact or it is very specific to lamb protein. So we introduced lamb again in this background, and you can see here the puncta were gone. That suggests that this phenotype was very specific to lamb deficiency. And this is the quantification when we mutate lamp with a different allele, we see a sharp increase in lysotracker staining, significant increase, and then it goes down, suppresses when we provide human lamp, which also suggests that Drosophila and human lamp can compensate, human lamp can compensate for Drosophila. So I have told you that lysotracker is known to be upregulated in autophagy when autophagy is induced. So we wanted to check whether autophagy is normal in lamb mutant or it is defective, because mammalian lamb is known to have defects in autophagy, like accumulation of autophagosome. So we use a sensor which is dual tagged, like GFP and RFP. So what happens, GFP protein is very sensitive to the acidic environment, and its fluorescence get quenched when there is like acidic environment. So we expect that there will be both red as well as green fluorescent in autophagosome, which is not acidic, while when autophagosome fuse with lysosome, it become acidic and it form autolysosome, and then there in autolysosome there will be no GFP signal, only M-Cherry signal will be seen. So if there is disturbance in autophagy, like we see more of autophagosome, more of green and red, both green and red, and less of only red. So in this way, we check autophagy. It's a usual reporter that we use in autophagy research. And what we found, you can see here that in fed condition, there is no activation of autophagy that was expected. And in starved condition, we can see nice panta. So when we mutate LAMP, like in 2 copies, and when is control LAMP heterozygous condition. So which indicate that we don't find a defect in autophagy in LAMP mutant, which was also surprising because mammalian LAMP is involved in autophagy. So we quantified it with the similar manner with the GFP and the red signal, green and the red signal, and there was non-significant change in the LAMP mutant. So we thought that if a macroautophagy is not affected, it could be possible that endosomal macroautophagy is affected, other form of autophagy. So we check, I already told you about the reporter we use, FARQ best reporter. So we checked EMI in the LAMP mutant and compared it with the control. And to our surprise, we found nothing, like there was no difference in the LAMP deficient and LAMP LAMP normal flies. So we quantified it and there was no change. And the autophagy was induced in response to starvation, but there was no change compared to control. So this was the result we found till now, and we found that their LAMP lead to acidification of endolysosomal compartment, LAMP mutant, but it has no defect in autophagy. So we try to check a lipid profiling in LAMP mutant because mammalian LAMP is known to be involved in trafficking of lipid and cholesterol mainly in lysosome, which is very important for the membrane structure. So what happened in Drosophila? Drosophila has do not have a closed circulatory system. It has an open circulatory system where it has hemolymph, which you can consider equivalent to blood, but it doesn't flow in closed artery or vein. It directly bathes the tissue. So when Drosophila have the diet, the lipid in the food is converted to diacylglycerol. So this diacylglycerol is a source of energy in adverse conditions. So what happened in the adverse condition, this diacylglycerol changes into diacylglycerol and transported to other tissue where uh, uh, fat body energy is needed. Like during flight, Drosophila need energy in flight muscles. So it is transported from fat body to muscles. So it is stored in the form of TAG, but it is not transported in the form of TAG. It is transported in the form of diacylglycerol with the help of apolipoprotein, which is not shown here. So when fly needs energy, this TAG will be converted into DAG and will be transported to other organs. So we try to quantify both TAG and DAG in Drosophila LAMP mutant and compared it with control flies. And what we found that DAG was accumulated, but there was no disturbance in TAG. So this led us, here you can see that LAMP mutant has elevated DAG. It was accumulated, which come back to normal when we have provide other copy of LAMP. So these results suggest us that there is no defect in lipid storage or in flies in LAMP mutant, but there is defect in transport of lipid. So lipid is not transported properly, it is accumulated. So this is a very interesting result because mammalian LAMP also has been manifested in defect in lipid transport. So it shows that Drosophila and mammalian lamp work similarly somehow. This work was done in the collaboration with the Noreen and Dr. McIntosh in our state university. So they did this experiment. So then we see cholesterol, because lamp is known to traffic cholesterol to the lysosomal membrane. We did a Philippine staining in lamp mutant and control, and what we found, there was a lot of Philippine accumulated in lamp mutant. So Philippine is an antibiotic from bacteria, and it is known to bind to cholesterol, and it changes fluorescence when we bind to cholesterol. So we measured the punctate structure intensity, and we found that lamb mutant has elevated cholesterol Philippine staining, indicating that there is a change in the cholesterol trafficking accumulation in lamb with lamb deficiency as compared to control. So these results suggest that there is a problem in cholesterol trafficking in lamb mutant. And it also suggests that in Drosophila and mammal, this process is conserved. And although we don't find any defect in autophagy in lamb mutant. So I'll summarize this result. What we found that when we considered lysosomal membrane, lysosome is like the acidic, it is lumen has acidic pH, and that is very crucial for the degradation because the acidic hydrolase act on a low pH. So this pH is maintained by two protein B-ATPase and TMEM 175. So what happened, B-ATPase is known to efflux proton, and TMAM is known to efflux proton. So in mammal, LAMP protein are known to oppose these two protein. So when LAMP is mutated in mammal, there is no effect in lysosomal pH, because the one which is involved in influx and efflux, and the one with the efflux, both are disturbed. So there is compensation, and overall result is no change in the pH. But what happened in Drosophila? In Drosophila, this enzyme, which efflux proton, is not present. So there is only VADPase and lamp protein, and we don't know exactly the function of lamp protein. So we hypothesize that when lamp protein is mutated in Drosophila, it affects pH of lysosomal membrane, and that's why lysosomal lumen, and that's why we see a lot of acidic structure in lamp mutant, because there is no TMEM present to compensate and efflux the proton. So we come to an important finding, and we publish our result in 2022, that flies, although do not, flies lamp are not required for viability, it has an important function, that is, it maintains the acidification of endolysosomal system. So when lamp is mutated, there is altered endolysosomal pH, and that can be a reason for lipid transport defect and cholesterol trafficking. It could be, I mean, we are not sure at this point, and one of the PhD student in my lab is working on this, how exactly this process is going on. Because this, as I already mentioned, the lipid transport and acidification is manifested in many disease conditions like Alzheimer's and Parkinson's. So I was interested in studying lamp protein other than tissues, other than fat body, like in the brain, mainly in the neuron, because I had a background from circadian rhythm. I did my first postdoc there. So I tried to convince my present boss that I will see lamp protein in brain. So I'm exploring this. but I'll show you some of the interesting results that we get. So when we say neurodegenerative diseases, the first thing come in the mind is like neurons are degenerated. So what happened in the healthy neurons? There is like no accumulation of aggregated proteins, or the uh, process of autophagy occur nicely, and it remove the accumulation. Since neurons are non-dividing, they cannot dilute their toxicity. So that time the cellular quality control is very important. But what happened in disease condition or in aged condition, there is like crowding of aggregated proteins and other waste, and autophagy process is also known to be decreased. So there is like a degeneration of neuron due to these toxic materials. And there are many diseases like Parkinson, Huntington, Scotty, which are known to be manifested with this decrease in autophagy and increased neurodegeneration. So how we use fly to do this? What we do is like we model human diseases in flies, because the gene responsible are mainly known. So we model this gene in fly, and we see what happened, whether fly can reproduce those phenotype, and if so, then we do genetics like screening of drugs, or we do genetic manipulation to treat these diseases. And the other good point is like in flies we can do a genome wide screening, a large level of screening, with very easily as compared to other model, mammalian model like mouse or a different model. So fly provide us a platform to study these diseases. So I can show you an example. Since I'm working on Parkinson disease, I'll show you an example. Parkinson disease in human has a accumulation of alpha-synuclein. Alpha-synuclein is a protein which, when is increased or mutated, there is a tendency to accumulate and form intracellular inclusion, which is also known as Levy bodies. So this Levy body is a accumulation of alpha-synuclein protein mainly, and then when these Levy bodies are present in neuron, it can lead to neurodegeneration. And you can see the in elderly people, mainly in age-related manner. So these people have motor problems like motion problems, they have tremors, and they have other indirect problem also like loss of olfaction and sleep disturbances. So what happened in flies, when we express these alpha-synuclein in flies, it reproduce the same phenotype, same uh, phenotype as of human. So you can see this is the fly brain. It shows an accumulation of Levy bodies, alpha-synuclein, and fly brain. So I will show you another process where we score how this accumulation has a significance on fly neurons, like similar way like human. So here what we do, that when we mechanically stimulate flies, they will with the mechanical disturbance, they will start climbing at the top of this tube. But when the flies have a defect in neurons, especially when the motor neurons are defective, they'll not climb it. So you can see here clearly. So these flies, so you can, sorry, you see that the flies, one which were normal can climb to the top of tube, but the other one which has a defect in I think LS, it was not climbing at all. So in this manner, we measure the climbing performance in the flies. So what we did, we measure lamb protein, lamb flies, like lamb deficient flies, and we thought we will see some defect. So this is the uh, um, process where we calculated the performance index, or you can say the climbing effect in a age dependent manner. So in young flies we don't see anything, And when we mutate lamp, we use two different lines. We found that there was no change. So fly were not different than the climbing defect. There was no climbing defect. So we thought to do this in the background of alpha-synuclein. So we express both alpha-synuclein and we mutated lamp. So what we found that when alpha-synuclein was alone mutated, and the we express the mutated form in a very specific neuron called dopaminergic neuron, which is responsible for the motor function in flies and in human. So we found that there was some defect. You can see this green line. So there was some defect which was age related in alpha-synuclein. But when we delete lamp in this background, what we found, there was significant change in the climbing ability in young, in not in young age, but in when the fly were getting older. So what does it suggest? That lamp itself has no defect, but when it comes in a sensitized background, like the expression of alpha-synuclein, it it elevate the I'll say toxicity of alpha-synuclein. So we use a mutated form of alpha-synuclein which aggregate more, which is known to aggregate more. And what we did next is we provide lamp with a promoter which expresses each and every cell. And we found a very remarkable thing, that when we mutate, when we express lamp, it can bring back the climbing abilities of flight. So this black line you can see, which has both alpha-synuclein mutated form expressed and in the lamp background, mutant background, and then we give exogenous lamp with tubulin. And we found that the flies were normal as compared to the wild type with no alpha-synuclein expressed. So it is very interesting because it not only the rescue the toxicity of alpha-synuclein, but it also bring back the flies to more like wild type, even better than the alpha-synuclein alone. So this suggests that lamp has a function in lamp might uh, decrease the severity of alpha-synuclein toxicity. So now we try to understand how it's doing so. So what I'm doing is like I stain Drosophila brain with alpha-synuclein and see what is happening to alpha-synuclein level. So for this, we use a simple Gal4U system that is routinely used in fly background. Sorry. And then I express human alpha-synuclein, and I did the antibody staining with the alpha-synuclein, and we use the driver which express only in neurons in adult, which is called as NSIB. So what happened, you can see here that alpha-synuclein is the right panel, the alpha-synuclein. We use both male and female, and surprisingly, we found that alpha-synuclein was not increased, while it was, in contrast, it was decreased in the lamb mutant. So that was also very surprisingly. But we know that for now, we know that there are a lot of research which tell us that alpha-synuclein is in, the toxic alpha-synuclein is in different form. It form a very small oligomers which cannot be detected, very hard to detect. And those are like very toxic, even than aggregated forms, Levy body, which called. So our result suggests that when lamp is present, it tends to keep alpha-synuclein in less toxic form. And when lamp is mutated, there are more of oligomers which are more toxic and which lead to clambin defect. So now we are trying to find out those oligomers, and we are using different staining to label those oligomers, but we are not succeeded yet. to label those oligomers, and we have published this result in 2022. So where does it lead us? So I showed you the function of LAMP protein and how LAMP works. So we come to know what LAMP do in Drosophila and what it don't do in Drosophila. So it is not required for viability. It is not required for autophagy and other form of autophagy, that is endosomal macroautophagy. So what is required for lipid assimilation. It is required for proper acidification of endolysosomal system. So, and it also enhance PD model in Drosophila, Parkinson's model. So now we are doing trying to find out how LAMP1 counteract alpha-synuclein toxicity, and what form of alpha-synuclein in the brain is present in the LAMP mutant. Mostly we think that it's oligomer form. And does it affect the pH environment of alpha-synuclein and hence affect its degradation? So these are the things I am trying to do now. And I got a grant from American Parkinson to study this part. So I'll not take more time, and I'll take a few minutes to acknowledge the institute and my lab members and the collaborators. So my PI is Andrea Jenny, and he's a very nice person, and he gave me opportunity to study on the like very challenging project, and he always helped me. And the other people like Anandita, Anna, Margot, they were like former lab members which were actively involved in the LAMP project. And their current member, Laurie, Carrion, Prasoon, and Jonathan. So they are studying different projects like autophagy in heart, and autophagy in like muscles, like that. And we have a collaboration with Dr. McIntosh's lab in Iowa State University, and our neighbor lab, Anna Maria Calvio lab, which is a very renowned lab in autophagy field, and with Serge Berman in Paris. And I also take this opportunity to express thanks to American Parkinson Association for providing me this research grant and the facilities at Einstein, like confocal EM, to make this study possible. And thank you all for your attention. And I'll happy to take any question. Thank you.
Thank you, ma'am. It was actually very informative, and we are grateful to have you with us. You have taken
No
our time
problem.
giving us this lecture. We'll begin with some questions now. And I would ask
Yes.
her here if they have certain questions, they would come up.
So good morning, ma'am. My name is Satya, and
Hi.
we do BTech file sharing. So there are two questions. So firstly, what are the diverse therapeutic approaches that are targeting the autophagy neurodegenerative disease that are encompassing the chaperone-mediated autophagy and the microautophagy, and how do they intend to modulate the autophagic process to bolster the neuronal survival and function? And the second question is, can you elaborate on the topic of endosomal sorting dysfunction and related to the LAMP?
The LAMP proteins. Okay. So your first, sorry, your voice was a little bit broken, but what I understand is you are asking how CMA, other form of autophagy, CMA and endosomal macroautophagy are affecting neurodegeneration, are manifested in neurodegenerative disease. That's what is your question? The first one.
I'm asking specific to, is there any diverse therapeutic approaches that are targeting this type of pathways?
Yeah, so CMA doesn't exist in flies, but it exists in mammal. So I know, according to my knowledge, there is like some drugs that my neighbor lab, Anna Maria Curbo lab, mainly they work. So they are targeting CMA with some drugs that can though they have used lamb protein also, lamb2A, that is absent in flies, but is present in mammals. So what they do is they increase the level of lambs. Lamb protein is known to decrease with age. And they think that CMA decrease with age because the lamb protein is decreased. So what they do is they overexpress lamb, and they can see some more clearance of aggregated protein and expansion of lifespan, and the less of aging, like that stuff. So, and I know some drugs that I really don't know the name, that they have established that can increase CMA activity, and then that can be beneficial for like protein clearance in neurodegenerative diseases like Parkinson's or Alzheimer or Huntington's. So what was your second question? Sorry, I really missed it.
Yes, ma'am. So, ma'am, can you elaborate on the topic of autosomal sorting dysfunctions related to the lab proteins?
Okay, so related to the lamb protein. So what happened in when I say in flies, lamb is not studied well. We are the lab which studying lamb, and we found that lamb protein, when lamb protein is dysfunctional, like deleted, like null allele, we don't find, we see the some of the sorting protein like cathepsin in our lab with Western blotting, and we found there were no change in cathepsin enzyme that is present in lysosome. And for mammalian lamb, according to my knowledge, there is no change according to my knowledge, but I have to go back and see. I don't have much knowledge for the mammalian lamb.
Thank you, ma'am, for it.
No. Thank you. Morning.
So my first question is, how much similarity is there between the mammal and lamb-1 protein and the fly lamb-1 protein, the organs?
In flies, okay, so there is like 56%, 54% of sequence similarity.
So if we were to transfer a fly lamb-1 protein to mammal or vice versa, would they act in the other organism?
Yeah. So what is your question in other organism? What I understood is you are asking that they can compensate for each other function. That's what you are asking?
Yes, yes.
Yeah, they can, because in flies we see that when we give human lamb, it can suppress the effect of phenotype of lamb mutant, but we have not done vice versa, like expressing fly protein to human. So it's difficult. So we haven't done, but that will be a very great experiment, and that we don't know. But yes, in flies, human lamb can suppress the defects.
I had one more confusion in one of the slides. It was written that the lamp-1 mutant reduces alpha-synuclein levels in flies.
Yes, yeah, it's reduces. So what we did is we saw in the brain of flies the neurons which are responsible, which accumulate alpha-synuclein. What we found that with alpha-synuclein antibody, there is less accumulation of alpha-synuclein. So that's what I was saying, that when we see alpha-synuclein, with the antibody, we can't see the oligomer, but because they are very small. And recent research suggests that alpha-synuclein has more toxic form in the form of oligomers, soluble oligomers. So they are soluble, it's hard to detect them. We are trying to do those with the MI tracker staining, but we have not succeeded it. So we think that there are more toxic form like oligomer accumulating in LAMP mutant. And LAMP in normal condition reduce those accumulations. So when the LAMP is not present, there are more accumulation of oligomer that we cannot see from antibody staining.
Ma'am, my doubt is autophagy is usually missing in cancer cells. So is there a change in the lab protein constitution or anything related about the cancer cells?
What is your question? Sorry, I could not hear you properly. What is your question?
Cells. Autophagy is usually missing in cancer cells. So is there a
Okay.
change in the constitution for those cells or is there a different change?
Okay, so autophagy, according to me, although I have not worked ever in cancer cell, autophagy is very like not clear. There is no clear concept of autophagy dysregulation in cancer cell. Sometimes it is beneficial for cancerous cell, and sometimes autophagy accumulation relieve cancer, it's kill cancer cell, I'll say. But autophagy has also been shown to be beneficial for cancer cell, and they provide energy to the cancerous cell. So although the research doesn't say clearly what autophagy do in cancer cell, but I haven't worked on cancer cells, though I don't know the detail.
Thank you.
No problem.
Thank you, ma'am, once again for such an informative lecture that you have given us.
I saw someone was asking some question. Okay.
Thank you, ma'am. The question was, many studies have shown that neuroinflammation due to aging also leads to neurodegeneration.
Right,
Was
right.
it considered when you are working on the toxicolor models, especially in the case of alpha-synuclein protein? So can we say the neuroinflammation also does play a crucial role in the dementia or healthy body dementia classification?
So you cannot see dementia in flies, but yes, of course, most of the phenotype in human can be reproducible in flies. So you can see the accumulation of alpha-synuclein in fly brain, and that is age dependent. So only older flies shows those accumulation, only older flies shows climbing defects. And when we say older flies, it's not like human. It's like since fly have a very less generation time, it is like 45 days is older. So we study aging and age-related neurodegeneration. It's very easy to study in fly because those kind of condition is in fly is only in 45 days as compared to human. When we say older is like 60 or 60, more than 65.
So, can we have any comparative analysis?
Yes, of course, we do those things. We study flies for aging and age-related neurodegeneration, Parkinson, Alzheimer, all those are studied in flies.
Is it supporting factor enough that we can say that neuroinflammation can be a good factor for all the studies that have been performed?
Sorry, I cannot hear your question. Your voice is like broken.
But how much can we use the statement that neuroinflammation and aging go hand in hand and in the progression of such neurodegenerative disorders?
Can you write down your question in chat? So really the voice is broken and I can only hear neurodegeneration and like that stuff. Not exactly the question.
How much can we consider the neuroinflammation as a supporting factor for such aggregation of proteins or neurodegeneration?
Oh my God, I'm so sorry, but seriously, your voice is so broken that I can guess the question only. I cannot understand anything you are asking. Maybe you can write it down in the chat and I can study it. No. No. Again, I mean the voice is uh, broken. I can't hear anything.
Is it audible now?
Yeah, now it's better.
Actually, we are typing the question and you'll get it soon.
Okay. Right.
And the question is in the chat box.
Is it the full question? It says that so like we can support the neuroinflammation as a factor. Yes, of course. There is neuroinflammation reported with alpha-synuclein overexpression. So we can support neuroinflammation as a factor for neurodegeneration. Ultimately, there is neuroinflammation.
So, hello, ma'am. Am I audible?
Right. And
So, ma'am, my question is, what is the correlation? So, ma'am, what is the correlation between Levy bodies, like the combination of Levy bodies and the alpha syndicate, when it went in the size in Parkinson disease, when it came from normal to mutant?
so the alpha-synuclein is not present in flies. Like this is not the gene which is present in fly. When from human, we are expressing it in flies to see how it affect the flies, like similar to human or not. But human alpha-synuclein is present and it has a function most probably in trafficking of protein. So it's not clear, function is not known, but in human alpha-synuclein. But what happened when there are mutation, there is more of alpha-synuclein. So when there is more of alpha-synuclein, they tends to aggregate, and they aggregate in a form like inclusion body, which is called as Levy body. So in flies, similar thing happen when we express alpha-synuclein, especially even wild type, but especially a mutated form which has more tendency to aggregate, that form Levy body in flies. So we can see that. And then when this Levy body is formed in the fly brain, it degenerate the neurons where they are formed. So yes, exactly it happens as in humans.
Ma'am, like in humans, when it when it went to rise in recognition of Levy bodies, only then the Parkinson disease will gonna happen.
Right. So in human, when we see the disease symptom, it's already so late. So that is a problem with the neurodegenerative diseases. So when we saw the symptoms, there is already full-blown disease uh, uh, progress in human. And in flies also the same thing happen. So when we don't see anything in younger flies, only in the older flies, when there is like aggregate and Levy's bodies are formed, there is a phenotype we see, the similar as of human.
Thank you so much. Thank you, ma'am. As
Thank
we
you.
are about this session, I would ask Mr. Suresh to please come up and deliver a vote of thanks.
Good morning. Great week is being celebrated at VIT Bhopal University. On this occasion, I would like to thank our Honorable Chancellor, Dr. G. Vishwanathan Sir, Vice President, Mr. Sankanathan Sir, respected Assistant Vice President, Ms. Kadambari S. Vishwanathan, for their guidance and leadership, which is helping us in organizing such impactful events. I would like to thank today's speaker, Dr. Satya Surabhi, staff scientist at the Albert Einstein College of Medicine in Bronx, New York, USA. In this science week, they will be having a lecture series where speakers hailing from all over the globe will speak in their line of expertise. I thank them for accepting our request to participate in this extended celebration week. In this continuation, I would like to extend my sincere thanks to the VIT Bhopal administration, deans, directors, faculty, colleagues, students, and all the participants of National Science Week. I would like to thank coordinator Dr. Saran Tripathi and co-coordinator Dr. Pradeep Kashyap and Dr. Ashok Kumar Bharat
for their continued support as the esteemed team members of organizing committee. The National Science Week celebration at VIT Bhopal is catalyzed and supported by the National Council for Science and Technology Communication, Department of Science and Technology, DST New Delhi, and Madhya Pradesh Council for Science and Technology, MPCOST. On behalf of VIT Bhopal, I thank NCSTC and MPCOST for giving us the opportunity to celebrate this for a week. Thank you.
Thank you all for joining us today.
Thank
We all
you.